Greetings and welcome back to COG 266, Brains, Minds, and Consciousness. Today we are going to carry on our discussion of neural function and talk about neural disruptions. Before we can talk about these disruptions, though, we should talk about, just recap the function, right? So, basic neural function with some nice gifs borrowed from Crash Course. Ions will flow in and out of ion channels to change the differential of the charges across the cell membrane. Inside the cell is some amount of ions, outside the cell are different ions, and the difference between these two creates potential. When enough potential is amassed in the cell body, it triggers a cascade that will make ion pumps all across the axon trade ions in and out, and then cause this change to move throughout the cell. We see a little blip of our friend Hank Green. So that's the idea. This is the action potential, the way that ions will, by moving ions in and out of the cell, we send the signal down. And then finally, at the end of the cell, neurotransmitters are emitted from the axon, from the terminal bulb of the axon, into the synaptic cleft, where they will trigger the next cell's dendrites, or sometimes just the membrane, to either let more ions in, or let less ions in, or let certain kinds of ions in, etc. Right? So that's the regular function. This is the standard function of neural cells, right? There's excitatory and inhibitory synaptic connections that will interact with the single neuron's membrane to help it alter its charge. Neuromodulators might also be in the mix, changing the way these functions happen, so maybe increasing sensitivity, etc. Once the neuron's charge crosses the threshold, it causes that cascade down the axon. That's the action potential. When that action potential hits the end of the axon, neurotransmitters and or neuromodulators will be released into the synaptic cleft, and also any given cell might respond differently to these different transmitters. And finally, and that also causes the nearby neurons to change their charge and potentially send new signals, right? And then finally, neurotransmitters are reabsorbed from the cleft into the cell, either in part or in whole, and you get it all back. I hope this cycle is starting to ring true for you, right? Neurotransmitters hit you. That might make you fire. You fire a few times. That might, overall, that you are sending signals down your axon. I'm calling you a single neuron, which then emits neurotransmitters to the next cells, right? This is the regular function. This is the kind we've been kind of harping on. And now let's talk about the many, many ways that this function, this relatively complicated process, can be screwed with, right? So there's two major categories. We'll talk primarily about the one antagonist, but we will also discuss agonists in treatments. So antagonists, these are the ones that are going to bind to and disrupt neurotransmitter sites, as well as some of them will affect specifically ion channels, not just neurotransmitter channels. And agonists, which are the things that actually mimic neurotransmitters for various purposes. Let's talk antagonists first. Antagonists are, there's often you'll see this as toxins, neurotoxins. There's a wide variety in the world, especially out there in nature, in that forever arms race of animals trying not to get got. The idea is that they will bind to receptor sites and prevent neurotransmitters from activating. Now that sounds fairly simple, but this has a lot of subtlety in what they choose to, choose is an anthropomorphizing term, what they are targeting, right? So neurotoxins might target very specific things. They might be more temporary or longer term. They might have pretty severe effects or less severe effects. So let's walk into a few of these and talk about what they do and why that's bad. So first, tetrodotoxin. This is what you might typically hear from the fugu fish. It is one of the most, if you've not heard of this is a very dangerous fish to try to consume because these toxins are readily released into the body if you don't cut them just right. So they have a popular life as a very fancy, very dangerous meal. You must really trust your chef and whatever. Very expensive. So yeah, don't mess with that. Don't screw around with that, because I'll tell you why. What tetrodotoxin does is it blocks incoming sodium channels, so positive activation, positive ions, which will raise the threshold for activation in the cell, right? So if it blocks those specific channels, it's going to make it so that the cell has a higher threshold for activation. Why is that bad for neural function? Well, think of it this way. Under normal circumstances, your neurons are kind of hitting this balance of every so often they will reach the threshold and they'll fire and they'll do their normal job. When they're tuned just right, they only really want to fire at that threshold. If you raise the threshold, that means it takes more excitatory activity for the cell to fire, meaning the cell becomes, in a way, sluggish. It's less responsive, and even if it does respond, it has lower firing rates when it does, and it may just cease to send any information at all, meaning it's really not doing its job, and it's not able to participate in the neural dance that is the interconnected network. On the other hand, chlorotoxin. Well, this is scorpion venom. It's a neurotoxin, just like the last one, and it's similar to tetrodotoxin, but instead it obstructs the incoming chlorine, negative ion channels. So what effect does that have? Well, quite the opposite. The lowered threshold shows that if it's going to take less excitatory activity to make the cell fire and also continue to fire, meaning this cell becomes hyperactive. It will fire when it's not supposed to, which is just as bad when you're dealing with a network. It becomes overly responsive, has these higher firing rates. It may cause a sort of overexertion, and it may also cause a certain amount of seizure-like, sending signals when you're not supposed to nearby cells. So that should, I hope, illuminate the idea here that we really want thresholds to be where they ought to be. You don't want them higher and you don't want them lower, right? So if it's too high, cells can struggle to function and can just fail, and they may, that may cause them to die off in one way. And if it's too low, the cells function way too much, causing excessive firing, and in addition to just sending signals where it shouldn't be. Remember that the cells are hungry, so it's wasteful and dangerous to the cell's internal metabolism to be firing constantly. These are both bad cases. Now, an interesting aside that you encounter a lot when you start to look into toxins is in cancer research, toxins can be used as a treatment. So they find that certain toxins do a good job selectively damaging cancerous tissues. Now, why would you want that? Well, remember that in cancer, the cancerous cells are typically, they're growing wildly in the body, disrupting other biological functions, disrupting everything, Cancer is a hell of a thing. And being able to disrupt the activities of those cells and the neural connections in particular can help to slow and kill tumors. Now, this is just one piece of the huge oncology puzzle out there in the world, but it's interesting to note that toxins can have very useful applied benefits. That's why actually you see on a couple of my slides here, we are milking a scorpion, so to speak. This is what it's called. You just get them to emit their toxins, and you can use them for other stuff. Usually not too harmful to them. You want to keep them happy so you can get more, right? So let's continue on our show of terrible things that happen to nerves. Clostridium botulinum, also known as botulism, it's a soil bacteria that thrives in low oxygen environments, and it emits a toxin that actually degrades the vesicles, preventing neurotransmitters from even releasing. Again, as we always are doing in this exercise, is figure out what's the problem, right? What is this going to do to the cells? Well, if I'm failing to release neurotransmitters, then essentially that means I can't communicate with the other cells, right? The cells around me cannot receive a signal from me because I don't emit enough neurotransmitter to trigger them, or perhaps less than average, right? So it's a little bit different than the preventing firing of the toxins that we saw. The blah, 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 tetrodotoxin and chlorotoxin. Because it's not just raising the threshold, it's kind of affecting the cell's ability to communicate at all, not just changing the way it communicates, right? Which, not to downplay how bad it is to change the way it communicates, but it's also, it can affect cells nearby because it's making it so that this one cannot tell anyone anything, right? So given that a cell might be, a single neuron might be connected to several other neurons, this is pretty bad, right? Another interesting one is Clostridium tetani, which is tetanus. Similar to botulism, it thrives in rusty environments. So you'll see this bacteria living on rusty nails. This is why the classic thoughts on tetanus is if you step on a rusty nail, you're going to have to get your tetanus shot, right? In a similar way to botulism, tetanus is going to degrade your vesicles, but it primarily affects inhibitory connections and not excitatory ones. That's a weird change. What can that do specifically? So if you have an inhibited response, a degraded inhibitory response, what can that cause? Well, what this means is that the neurons, in these affected areas, they can easily be triggered to fire, but they can't as easily be regulated by the other connections that are kind of control how much they fire. So all activity that says they want, hey, fire, like that's getting through just fine, but other signals that are trying to limit that rate can't get through. Now, if you've ever had a Charlie horse, you know what this feels like. The neurons activating your muscles are perfectly happy to fire. They say, yeah, cool, constrict, this is great. Whereas the ones that say, hey, hold on, we don't need to constrict anymore, aren't getting through. And this is, in a Charlie horse, this is typically a small scale thing caused by, oftentimes it's like you're not getting enough electrolytes, right? Certain kinds of salt. So it's not a sign that you've been bitten or you've got tetanus. But this is the same kind of thing that tetanus causes. In fact, it's known as lockjaw when you get when tetanus causes this kind of activity in your mouth area. So it can cause this, my mouth is happy to open, but it's stuck because the inhibitory signals aren't getting through. So you see, there is subtlety to the kinds of issues and maladies we can acquire. Let's look at an interesting one from history. Curare. This is a plant-based inhibitor, so we're not in the insect and bacterial zone anymore, that will typically bind to neuromuscular synapses, temporarily preventing their activation. Now, this was used historically to create paralyzing coatings, and that's kind of spoiling what this is. What is neuromuscular inhibition going to do? Well, that's quite simply paralysis. So if this antagonist is preventing musculature neurons from receiving messages, that means they cannot activate. They can't constrict. They can't do anything. They are blocked up, and the mail is not getting through, right? So the muscle can't contract. It can't do anything. Hey, we're dunskies. Now it's shorter term than some other ones, such as cobra venoms, a bungarotoxin. I don't always know the names of these things, but pronunciation-wise, but I give it a shot. Cobra and other snake venoms, but cobra is the one that we know most about. It's irreversibly binds to the neuromuscular synapses, similar to the last one, but this means it's permanent. It permanently destroys them. So similar to curare, but forever. So not to say that, so this is a similar action to curare, but because it's so permanent, because it's so insidious, that this is what it will prevent you from ever sending these signals ever again. That's why cobra venom is so dangerous, is that it just doesn't come off. So you might recover, but the cells that it touched will not. And another element here that's just curiosity is like, that's a pretty sophisticated biological weapon there. It sends this out and says, hey, I've made this special chemical just for your musculature that destroys it. That's pretty wild. So that's an array of antagonists, a lot of ways that we can screw with neurotransmitter sites to affect their function. They're all related, right? They're all doing the same kind of thing. They are messing up ins and outs and different activities that the cell needs to do to be able to send normal activations, right? But some of these are more subtle, just like tetanus targeting only the inhibitory connections. Strange things that happen to us. Hopefully we can avoid them. So this brings us to agonists. These are drugs that will mimic the activity of neurotransmitters. So this will cause cells to activate without the neural activity contributing. So you can send neurotransmitters in to promote activation in areas that may not be getting it. So there's a huge variety of these, and they can affect different neural pathways in complex ways. You know, this is the state of modern neural medicine, is using agonists to help regulate neurotransmitters that might not be working well in the brain. And so there's some more on this in the next section, because we're going to talk about how they're used to treat certain kinds of longer-term disruptions. So we just looked at a lot of acute specific substances that are going to trigger neurological dysfunction, right? Trigger? Huh. But some disruptions are longer term, right? These are the things that we think of as diseases, right? So in a disease, specific kinds of neurotransmitters and modulators are the real concern, because it seems like there is some broad dysfunction across larger parts of the brain that make it hard to engage in certain activities. So some neural populations are going to be outsized effects from others. Let's start with Parkinson's. So Parkinson's disease is a progressive degenerative motor disorder. So the general idea of that progressive means that it continues to get worse. And so patients will develop things like tremors. Their hands will shake, right? Their arms will shake. This is a classic thing. If you've seen anybody with Parkinson's, it's a sort of a telltale sign. So their motor activity becomes slow, difficult to start, and rigid. We can look at a little example of sort of how these things can be diagnosed. So in healthy control patients, you see stepping patterns that have this characteristic kind of stride length. The strides will be, they were kind of cyclical in nature. They ebb and flow. You will move faster and slower, and they will coordinate with each other. You may vary in your overall speed and the length of the strides you take, but your strides kind of go together. In Parkinson's patients, though, you'll see a lot more variability. The strides are not coordinated. They're not really following an overall pattern. They are a little bit more wobbly. And wobbly is a kind of, okay, that's not a fair term. More like they have a lot more variability, and they aren't being regulated together in a way. So the movement variability is larger, and it's less structured. It's at least structured differently. They're having to rely on some other structure to succeed, because their normal patterns, like those in healthy controls, are not available. So Parkinson's disease is caused by reduced dopamine levels, specifically in the basal ganglia. This is an area of the brain known for coordinating voluntary movements. Voluntary meaning things that you are trying to do, intentionally doing. So the reduction of that dopamine in that area means that the function of this brain region isn't really working so well. How do we treat it? So a drug called L-DOPA It passes through the blood-brain barrier up to the brain and gets converted into dopamine. So it actually becomes dopamine. Newer treatments since then, since the development of L-Dopa, involve agonists, so things that will just directly hit the dopamine channels instead of creating new dopamine, and also adding new dopamine producing cells. That's pretty powerful, but it's also a more experimental process that is a lot more difficult and expensive. So, but you could see how that would help. Having a cell that is able to produce this stuff in the brain without any outside influence, pretty useful. So, the drop off in dopamine, which is primarily this neuromodulator, rather, in one area of the brain affects the normal function of that area. So it makes it more difficult for the neurons to activate properly, leading to those tremors and activation issues. So the idea of treating this is try to reintroduce dopamine. It seems like not enough is getting there, so introduce dopamine or dopamine agonists into the brain. Hey, please, hey brain, keep trying. Hey, you're not getting all this stuff that you need. I'm going to hand you a bunch and see, hope you can successfully continue operation. On the other side of this same neuromodulator, dopamine, is schizophrenia. Schizophrenia is a mental disorder characterized by delusions, hallucinations, impaired judgment, and related disorders like anxiety and depression go along for the ride because it's a pretty widespread issue in the brain. And we don't fully understand this disease yet. There's causes are still being investigated. For example, the dopamine hypothesis is that it's actually excessive dopamine, or maybe too many dopaminergic receptors that receive, the same amount of dopamine. That may be the issue. It may be causing this dysfunction, allowing too much activity of some kind of neural circuit, some kind of neural population that ought to be activated like this. So drugs that raise dopamine levels can induce symptoms similar to schizophrenia, is the first piece of evidence for this. And successful antipsychotic drugs typically suppress dopamine levels. So this is another bit of evidence. And in animal models with increased dopaminergic receptors, as we've hypothesized in the top, they will exhibit these same symptoms. So the idea here is that too much of a good thing for, say, a person with Parkinson's can actually lead to issues on a different end of this spectrum of, oops, I've got too much dopamine activity happening. That's not great for my mental health. So dopamine is this sort of balancing point. It's this neurotransmitter that's obviously very key to the way our brains work, but too much treatment in either direction can activate symptoms of each of these two diseases, right? So sometimes it's so severe that you have to discontinue treatment. So high dopamine can be associated with these delusions, hallucinations, impaired judgment, while low dopamine can be associated with slow and difficult motor control and tremors. So if you have either of these diseases, you are attempting to pull yourself back towards middle. So from Parkinson's, you might take L-Dopa or other dopamine agonists, and in schizophrenia, you're going to take antipsychotics, which will bring your dopamine back in line. Let's talk about depression. This is a very common disorder, often characterized in part or whole as this persistent feeling of lack of motivation, difficulty experiencing emotion, difficulty deriving joy from life. This may be increasingly familiar. Hang on there. We're all in it together here. And it occurs at multiple levels. Sometimes it's a symptom of a different disorder. Sometimes it's like a major long-term life situation, or maybe it's a shorter-term bout of depression. But altogether, it is a fairly common experience, and it's even though it's one of these diseases that you may often hear as like, oh, it's just kind of like sadness. It's actually it has a fairly typical shape. This lack of motivation, lack of emotion, lack of joy kind of stuff. So it's thought to be related to your serotonin levels. Now, there's a history to this that's kind of interesting, how we discovered such a thing. So, Reciprine is a drug initially used to treat hypertension, which is just high blood pressure. The idea is, whoa, this heart is like beating way too hard. I'm going to reduce the neurotransmitter in the peripheral synapses controlling heart rate, and ta-da, the heart beats less hard. Cool. Okay. Oops. It caused depression in our patients. It's weird. You wouldn't think those things were related at all. This is one of the things to consider when you think about neural treatments, is that we're really just kind of swinging the hammer and saying, hey, I'm going to screw with your serotonin, and that might help your blood pressure, and it might cause your depression. So, but on the other side of things, other drugs that were used to treat tuberculosis, these would inhibit the production of enzymes that break down monoamines, of which serotonin is one, meaning there's more of them. It will keep more of them in the system, meaning they don't disappear as easily. They can continue to activate and be used to activate. So using this drug seemed to actually alleviate depression by increasing all monoamine levels, including serotonin, which is the thing that we sort of zoned in on as the correct one. So you don't really want enzyme inhibitors in general. It's a pretty heavy hammer to swing. And you couldn't use these same medications designed for tuberculosis for severe depression. It wasn't enough. So it was more than just the enzyme that was causing this issue. So they began developing drugs that were designed to attack the transporters that reuptake monoamines. Now remember, the reuptake transporters we've talked about is how the cells get the neurotransmitter back, right? So more of these monoamines would stay in the synaptic cleft and continue to help activate the nearby cells. So early versions of depression treatment inhibited all monoamine reuptake. Then they designed ones that would target specific monoamines. Finally, the ones that were most helpful were when you would target serotonin. This is how we come to SSRIs, which you may have heard of, as selective serotonin reuptake inhibitors. Essentially saying, hey, this one neurotransmitter seems most related to depression. I'm going to make it so it stays around a little longer and doesn't get trapped inside of the presynaptic neuron. It's going to stick around so you can maybe have better activations overall. So, treating depression is not fully solved, but you can see how techniques have developed over time, right? The more specific drug mechanisms were developed right along this line, and they're cool because they have very few side effects. It's not like I'm swinging the hammer to change all of the monoamines in my entire neural system. You don't necessarily want that. Hey, there's schizophrenia, right? But they're harder to develop because all these neurotransmitters are related, and it's kind of tricky to develop things that target just these one areas. But this is what's so cool about this continuing development, is that we come up with these things. Now, let's talk a minute about neuropeptides. These are large, complex substances that they're wildly different, and they're everywhere in the body. These are sort of our own tools that can help to trigger neuromodulators, and other such things in these same capacities. So, for example, some parts of your body will release neuropeptides to trigger neural changes in the pituitary system and the digestive system. So, meaning digestion and puberty are two complex processes that require different kinds of neural activity that the body can trigger when needed. So that's what these neuropeptides are all about. So in any given brain area, there aren't many neurons that are super sensitive to peptides and neuropeptides. But when they are used, they cause widespread processes. So not terribly well understood, but broadly, the thalamic systems will help to deal with metabolic regulation. So, things like how and where energy is getting spent. Neurons can help with that. Pituitary system, so process associated with your growth. You might need a different set of neural activations to handle the oddities of growth than otherwise. And the digestive system, so we're eating, stop doing that or we'll cramp up, right? Little things like this. So ways that our system can communicate with itself very broadly to say, hey, I need a whole bunch of change in the way that we deal with neurotransmitters and neural activation in a lot of ways. The way I will trigger this information is by sending these big spindly boys up somewhere. Now, you're not gonna memorize these things. This is just to show these are the kinds of proteins and molecules that will activate this stuff, and it's complex. Go take a neurobiology course if you want to know a lot more. But that's the idea. So we are able to send these around internally that to be able to handle some interesting phenomena. Now, there's one interesting class of neuropeptides that is pretty well understood. It's called enkephalins, or what is known as endogenous opiates. So exogenous opiates are like morphine or heroin or things like that are used for pain relief and other medical issues. Heroin actually used to be used for pain relief, but it caught on in other ways. So careful with that. So the fact that these work suggests, an opiate is a particular kind of molecule. And the fact that it does work means there's got to be some kind of receptor in the brain that's taking these things on, right? It's not specifically, like a serotonin agonist or anything like that. It's its own kind of chemical, and it's connecting, perhaps not chemical, molecule, right? And it's able to activate something in the brain that will inhibit pain. That's odd when you start thinking about this from the standpoint of, well, wait, which neurotransmitters is it screwing with? It's not. It is an active neuropeptide. So there's been an interesting study to investigate, hey, are there endogenous opioids? Are we doing this internally and not just externally from morphine and things like it, but internally? So if opioids are effective and we can identify their receptors, there must be some process that produces them inside our bodies. Otherwise, we wouldn't have those receptors in the first place. Why would we have the ability to interface with something that was so far out of our normal diet and routine and such? So what might cause that? What might cause the activation and the production of these and release of these in our body? So here is a set of subjects in this study. A bunch of people had their wisdom teeth out. One group was given morphine. One group was given sugar pill. One group was given nothing. This might be familiar. The placebo effect. So the sugar pill group, they are receiving something, but they are not actually receiving an active drug. Okay. What's going on? What happened in this study? Both the morphine and placebo groups experienced lower pain levels. This is that placebo effect. It's kind of well known, generally. But when the placebo group was given naloxone, a drug specifically designed to block opioid receptors, they experienced similar pain to controls. Endogenous opioid production may actually be a critical element of this placebo effect. The ability to produce this thing because you have been told that you can, right? Because you're being told that you are able to you will soon be, healthier than you were, right? This sort of optimistic process takes over. Tough to pinpoint. We still don't know exactly how the placebo effect works, but we're getting there with these kinds of studies. There's actually other evidence too, interesting stuff. Evidence from animal studies says that acupuncture for pain relief may also be actually producing these endogenous opioids. In addition, naloxone will inhibit these same effects. So the idea is, hey, a lot of people think of things like acupuncture as like, oh, that's just, weirdo medicine. It's doing something. It's somehow these very, very tiny pinpricks are activating opioid production internally. Hey, interesting. So, also don't worry, that dog is perfectly happy. Those little pinpricks do not hurt. In fact, if any of you have had the vaccine, then you will know that you don't always feel it. Right? So, this is the idea. The current thought is that this is a natural pain mitigation system, right? The fact that we produce these opioids internally somehow means that this is what it's there for, right? And it seems like there's a lot of ways to trigger it. There's simple intentions from placebos, like, oh, well, I'm going to get better, then that's fine. But also small stimulations without fear of placebo effects. A dog doesn't know it's getting a sugar pill, right? It does know, maybe not even very much, but you can still produce this change, these endogenous opioids, with these very small pinprick stimulations. So it leads to interesting questions about massage or acupuncture and endogenous opioids. This is how you start to bridge gaps. Western medicine is all attempts to be very science-based, but oftentimes it pulls things out of other alternative medicine that just makes sense. Aspirin was a tree root, and we said, whoa, or no, it's a bark rather. Yeah, either way. Important things can be found in places that we don't typically think of. So, get off your high horse, Western medicine. We got a lot to learn. I mean, also don't be taken in by. It's tough. It's tough out there. There's a lot of good medicine. There's a lot of crap medicine. Try to thread the needle, guys. There's a lot of life left for you. So, overall, neural diseases. I hope this is showing you that brain chemistry is incredibly complex, and we are really only at the beginning of our understanding. So, despite the prevalence of drug therapies treating all these different kinds of disorders, we are still working at targeting better and being more precise and developing all the techniques and the understanding of how different parts of the brain and the whole neural system interact in these ways. So we have methods for improvement, these selective inhibitors, but the research is difficult and it takes time. So that's neural communication in general. I hope it's all hanging together for you. We looked at how the physics and the chemistry of neurons will allow their membranes to transmit these electrical impulses. We saw how information moves across the synapse in neurotransmitters and modulators. We saw a little bit of a horror show of all the ways that synaptic activity can be influenced by external chemicals, but also how longer-term diseases manifest and are treated in the same domain. That's it for this little unit. As always, a final thought. Consider selective serotonin reuptake inhibitors, SSRIs. These represent a development to more carefully select which neurotransmitters we're targeting with our treatments. Given what we've discussed about neural function, what other advances in selective treatments could we strive for? What might be the pitfalls in implementing these new treatments in terms of unintentionally affecting neural behavior? All right, thanks. That's it. Take care.